So the way I do research is studying bacteria with microscopy. And it's my absolute pleasure to be in the Netherlands, where the whole field actually started. And it started by Anthony van Leeuwenhoek with a microscope just like this. So he made these microscopes actually to test fabric, the quality of his own fabric. And then he was curious, and he started investigating other things, water from the pond. This is a sample from the human mouth. And he could already see bacteria. And the resolution is not very high, as you can see. It's over 300 years ago. Now I show you a picture with a very modern Nikon high-end microscope. And this is actually a human cell, a HeLa cell here, with a nucleus. And here we have bacteria. So after 300 years, bacteria is still a challenge for light microscopy. They're very small. So what I'm doing is I'm applying electron microscopy because we have a much higher resolution. And we can get down to atomic resolution. This is a little movie, I don't know if it's playing. And this is not, it's a material. It's a gold particle. And I hope you can see these layer lines. These are different individual gold atom layers. So we can resolve atomic detail with an electron microscope. Now I tell you this, and then you open your biology textbook and look at the samples that people have imaged with an electron microscope, and you see samples like this. So this is certainly not atomic detail. This is a Caulobacter crescentus bacterial cell, and basically the only thing you can resolve is several layers of the cell membrane and the cell walls, the cell wall. But inside the cytoplasm, there is really not much detail left. So why is that? So the reason for that is we have to take our biological specimen and put it in the vacuum of an electron microscope. And to do that, we have to remove all the water in the sample. And it's illustrated here. If you have like this beautiful potato with all these beautiful features on the outside, when we dehydrate the sample, we plastic embed it, we stain it, we section it. By the end, we end up like something like a heap of potato chips. Also material then. So what I'm doing is I'm skipping all these traditional preparation steps that people have used for the last 80 years. And what we do is we take the bacterial culture and we apply it to an EM sample grid here. It's just a drop of liquid culture. So the only thing we do is we blot away excess liquid with filter paper. So we have a very thin liquid film on this EM sample, and we shoot it into liquid ethane. And the freezing of the sample is so fast that not even water molecules have time to crystallize. So the sample is vitrified, and the sample is not perturbed by stains or dehydration or plastic event or anything. But the second thing we do is we not only prevent artifacts from sample preparation, but we also do three-dimensional structure analysis. And how we do that, we take our specimen, here Mickey Mouse, we put it in the column of the electron microscope, and we tilt the sample. And we take a whole series of two-dimensional projections while the sample is tilting. And then we take the whole set of 2D projections, and in the computer, back project it to get the three-dimensional volume. For that, a big computer is really nice for me.
Where's the cheap computer?
Well, for me, that's my laptop does. Anyway, so we have really modern electron microscopes here at the IVL, at the Neeson Center, and here is an image of the microscope I just showed you in front of one of these machines that we use over at the Neeson. So it's a Titan Krios FBI machine. It's a very big machine to look at very small samples. Now here's the comparison of the sample I showed you before, a Caulobacter cell. This is the same specimen, not the same specimen, but the same cell type, imaged by tomography at the Neeson Center. And here now you can see all kinds of structural detail. We have an outer surface layer. If you watch the movie through, you can see this crystalline nature of the surface layer that's coating the cells. Then we have two membranes of a gram-negative bacterium in the cell wall. You can see the ribosomes, filaments. So now we have a very powerful tool to actually look into the three-dimensional structure of bacterial cells. And here is just a summary movie of what you can do. And this is a Bedell cell. It's a different species. And here in colors, we can see the membranes, the ribosomes, pili, chemoreceptor arrays, secretion pores, tubes, the chromosome. So now we can address all kinds of different questions using this technique. So now at the beginning I told you I'm interested in the bacterial behavior, and the behavior is called chemotaxis. It allows the cells to find nutrient sources in the environment. So this is E. coli on a slide. There's a sugar source down here at the bottom, and you can, if you watch the cells, you can see they all accumulate down here. So they can sense the sugar and guide their movement toward it. So here is so how they do it. These are fluorescently tagged flagella. So this is E. coli again. E. coli has about 8 to 10 different flagella. And when all the flagella motors spin in the counterclockwise direction, the flagella form a superbundle, and the cells swim smoothly forward. Now, if one or more of these motors start turning clockwise, the flagella bundle dissolves, the cells stop and tumble around until all the motors go in the counterclockwise direction again, and the cells swim smoothly again. And by controlling the duration and frequency of these run and tumble phases, the cells can direct their movements. Now, this behavior is based on two macromolecular machines. One is the chemoreceptor array. I'll talk about to you a little bit more, this here, and then the flagella motors here. And then in between we have a shuttling protein, QY, which gets the input from the chemoreceptor array, and when it's activated with the phosphoryl group, it goes to the flagella motor and changes the direction of motor rotation. So now, the rest of the talk, I will talk a little bit about this machine. So we have chemoreceptors, so they're called here MCPs, inserted in the membrane, and there are thousands, up to hundreds of thousands of these receptors clustered at the cell pole. Then there is a KW, which is it's a linking protein that helps link this array together. And then there is an enzyme, PA, that transfers the phosphoryl group to the shuttling protein in the presence or absence of the stimuli. So how does that look in the tomogram? So these chemoreceptors are present in all prokaryotes. They're in bacteria and also in archaea. So here's an example in an archaea cell. Is a methanogen. So it has a very interesting cell structure. It has a sheath and a surface layer and a membrane. And here we have such a chemoreceptor array inside you. So you can see the receptors inserted in the membrane. And down here, this extra layer is where the enzyme and the linking protein bind. Now if I start this moving, it will go toward the membrane. And then you will see the hexagonal ordered packing of these receptors here. So in case you have a hard time seeing this, I'll have some still frames. So here's the side view. Here we see here's the membrane. And here are the portions of the receptors that stick out outside into the periplasmic region where the attracting chemicals bind the receptors. And we have the receptors here. And here again is where the enzyme and the linking protein bind. So if you imagine you have a mug of beer, you drink it out, you turn it down, and you look down onto the bottom of your glass of beer, slice through where the linking protein and the enzyme sits, then you can see how incredibly ordered these receptors are packed at the pole. So here you will see it a little better in the next slide. So now I'm going to show you a movie. So this is not E. coli, this is a Salmonella mini cell. So here we have the inner membrane. You'll see a flagella coming out here, and then this chemoreceptor array. But the signal-to-noise ratio is very low. So if you're not used to looking at these gray images, you can see it's ordered. But to get a better signal-to-noise ratio, we apply a trick. We cut out individual volumes of our big volume, and then we take these individual volumes and we average them together. And that way we get rid of most of the noise. And we get a very clear view. This is about 2 nanometer resolution. We can actually fit the crystal structures in and can exactly tell how the receptors are packed. And here we see these receptors are packed as trimers or dimers. They form this hexagonal array. And here 2 dimers of one trimer face 2 dimers of another trimer. So in 2012, we finally teamed up with a crystallographer, and we combined our efforts with his crystal structures and our tomography data, and we finally could solve the whole array structure, exactly how these, the enzyme and the linking protein form rings and cluster these receptors together in these incredibly large and ordered arrays. So now, I told you in the beginning, I'm here in the material session, and I'm telling you about the bacterial behavior. But the beauty about fundamental science is it has implications for a lot of fields. And another session I could have spoken in would have been the health session, because for bacteria, for pathogens, a lot of times the first step of host invasion is actually a chemotactic behavior of the cells. But for materials, one possible implication would be, for example, living materials. It's been in the news from MIT very recently. They used E. coli cells coated with gold particles, and they could grow this living material. And they can conduct electricity and emit light. So chemotaxis could be used to direct the cells where you want to have material grow, and you can also regenerate material if your material is damaged. So I think it has huge impacts also for the materials field in the future. And with that, I just want to end and thank the group where I've worked in for the last 11 years before I came here to Leiden. It's the Jensen Lab at Caltech. And this is actually the picture of my goodbye party in November, right before I came. And I thank you for your attention.